but it's the only case that we have seen. This means that maybe we have overlooked cases before. I don't know, retrospective studies will tell us. But it looks like that PCV3 is a pathogen, can cause disease, but we do not really know which is the frequency in which this disease is caused.
Hello, I'm Peter Best. Welcome to Meet the Expert, the series on swine health management in practice, brought to you by Boehringer Ingelheim. In this episode, we're going to discuss porcine circoviruses, including a form that's only been described in the last few years for the first time, PCV3. For expertise on this question, we're looking to Professor Joachim Segalés, the veterinary pathologist and specialist in swine diseases, who is professor in the Department of Animal Health and Anatomy in the Faculty of Veterinary Medicine at the UAB University in Barcelona, and a researcher at Spain's CRESA Center for Animal Health Research. Professor Segalés, welcome and thank you for talking to us on Meet the Expert.
Thank you very much, Peter.
I have to say, circoviruses in pigs, suddenly there seem to be more of them than I thought. For some years I thought PCV1 occurred commonly on farms without causing disease, and PCV2 was the cause of this post-weaning multisystemic wasting syndrome, PMWS. But over the last few years we've now got this PCV3, and can we say with confidence that we got 3 species of the porcine circovirus circulating on swine farms internationally, or are there more than 3?
Well, definitively there's more than 3. PCV2, as you indicated, it was discovered as a cell contaminant in the PK15 cells a long time ago. It was back to the 70s and the 80s. And suddenly it was observed that the pigs had antibodies against that particular virus, but no disease associated. So it was soon considered that what we nowadays call PCV21, it was non-pathogenic for swine. As you perfectly indicated, PCV2 was discovered much lately, but was by the end of the 90s, but it was linked to clear disease, the so-called post-weaning multisystemic wasting syndrome, nowadays PCV2 systemic disease, in which since then it has been one of the most important diseases in swine. Even it's very well controlled by means of vaccination. But by 2015, and using a very interesting technique, which is next generation sequencing, that allows sequencing previously unknown sequences of viruses or even other pathogens, we discovered the existence of PCV3. It was in the United States, but when we developed techniques, especially molecular biology techniques like PCR, to detect the PCV3, we soon realized that PCV3 is distributed all over the world. Amazingly, a couple of years ago, in China, it was discovered PCV4, type 4, and this type 4, it's far away from PCV2 and PCV1, especially, and also PCV3. So it's a clearly distinct species of PCV. However, we tried to find it in Spain and in Italy, together with one colleague from the University of Padua, Dr. Giovanni Franzo, and we were unable to find PCV4 in Spain and Italy. However, the set of samples examined was limited. However, it looks like that in China it has been detected several times, and even in some other Asian countries. So it's very likely that if we are looking for enthusiasm, we will find PCV4 in other parts of the world. And this means that probably in the future, other PCVs may be found as well, especially when using techniques like next generation sequencing. So my guess is that probably PCV4 will not be the last PCV that we know nowadays.
We got better at detecting these things, and we're finding more because we're better at finding them. Yes, okay, I'm with you so far. And then, of course, diversity is a word with circoviruses in pigs, isn't it? Because, you know, as you've been describing to me in another conversation, PCV2 exists in a variety of genotypes, and they sort of compete with one another to be the dominant form. So just round up for me, how many, we got 4 known species. Now, forgive me, I'm bad at the difference between species, genotype, variants, subgroups, all these other things. But as far as I can say, there's 4 species, and then within the one called PCV2, there is a range of genotypes. So that would be, how many genotypes in PCV2 do we know today?
Yeah, you are completely right. We have 4 species of PCV, and within PCV2 we have at least proposed 9 different genotypes, from the letter A until letter I. And in fact, in PCV3, it has been so far proposed PCV2 or PCV3A, but there's some suspicions of a potential PCV3B. So, however, the big difference with PCV3 is that it's a much slower virus compared to PCV2. So the mutation rate of PCV3 is much lower than PCV2, and even PCV1 has also a low mutation rate compared to PCV2. So far PCV2 is the most variable PCV that we know so far.
And they're not closely related, or did I misunderstand you? I know, again, sorry, but I thought the PCV2 was only 60% genetically related to PCV1, which I don't know whether that's still the sort of level that we're talking about, which isn't very close relationship, and these others are not genetically related to each other anymore, to PCV2. They're quite distinct, are they? So have they occurred as a result of mutation of PCV2, for example, or have they occurred separately? Have they emerged separately?
Well, in fact, you are right. PCV2 and PCV1 are fairly similar, but fairly similar it means at the amino acid level of 70%, no more than that. But if you compare with PCV3 and PCV2, this similarity is of 40%, is even much lower. In fact, some phylogenetic studies propose that PCV1 and PCV2 had a common ancestor. And common ancestor, it means that might be 100 years ago, because it has been more or less timed in 100 years, and start with a diverging kind of evolution. It's very likely that with PCV3, it could have been even in a different ancestor, or even a much older ancestor. Yeah. So from this point of view, the relationship with them at the very end is basically because they have a very similar molecular organization, and as well, all of them, of course, are circular DNA viruses. Even the length in terms of nucleotides is different, or relatively different, especially when you are talking about PCV3 respect PCV1 and PCV2, which are much similar in terms of number of nucleotides as well. So it's clear that there's a divergent number of PCVs, and for the reason is what I told you before. I believe new ones will appear in the future, because every day we are much more fine in terms of sequencing, in terms of knowing the unknown, which is what next generation sequencing provides to a certain degree. So this is very likely that will happen. However, to trace back the most common ancestor of those viruses could be a very difficult task.
I'm going to then pursue the diversity a little bit longer. I'm going back to the genotypes of PCV2 initially. Does this diversity of PCV2 genotypes, is it something seen everywhere in the world simultaneously, everywhere the pigs are produced, or only in certain locations or at different times? So are these things sort of naturally occurring simultaneously?
Well, it's difficult to answer this particular question, because the different PCVs can be found in a given country, or even in a given region, or in a given farm even at the same time. However, by terms of frequency, PCV2A, B, and D are the most frequent one, and if you have to top them, the type D is number one nowadays in most parts of the world, not in all. In some, PCV2B is still the most frequent one, but after D there's B and finally A. And then the other genotypes so far have been found to a much lower frequency. This means, and we know since at least more than 20 years, that one single pig can be infected with two genotypes, two different genotypes. And this could happen in one single pig. And this is the reason why we can get recombinants of PCVs, recombinants between A and B, A and D, B and D, and others. And even we know that under field conditions, even very limited, certain recombinant forms may be detected as well. However, anyway, those three genotypes are the most frequent ones. And this is what it can be expected in the future. If you are going to a particular farm and you try to sequence the viruses found there, you may find whatever of them, because it's one particular farm, and then it's very difficult to predict which particular genotype will be found. But even it would not be surprising in one single farm to find the three major genotypes. This could be perfectly possible. As long as you are looking widely in the farm, you may find them.
Now, you've indicated to me that vaccination at the moment, PCV2 vaccination, is being effective against the genotypes that we're meeting today. Would there be any chance that it could also be protective against other circovirus species, or is that impossible?
Based on the similarity between the different PCV species, probably this is not feasible. So PCV2 vaccines protect against the existing PCV2 genotypes, but will not protect against PCV3, for example, or PCV4, and even with PCV1. Might be it could be some more cross immunity with PCV1 because are the closest ones, but probably would not protect. But anyway, PCV1 is considered non-pathogenic, presumably. So from this point of view, do not expect protection against PCV3, which would be the most important one, probably.
Let me pause for a moment to remind everyone that more information on this conversation, like articles, publications and videos, can be found on the website pers.com. Now, you said in PCV2 that we're looking at, so far, two genotype shifts. One before the year 2000, I think, where, therefore, before we had PCV2 vaccines, where I think the shift was it from A to B or some, I think so. And then in about 2012, you had the second shift in the vaccine area, and that shift was towards this dominant form 2D. Can I ask you, what are the drivers of genotype shift? You're a veterinary pathologist, but I'd like you to just explain to me how do genotypes compete, and how does one become the dominant one?
Well, I would enjoy having a good answer for that question, Peter, but I'm afraid I don't have it.
Sorry.
Because as we have been discussing, from an epidemiological point of view, those two genotype shifts, understood them as a global change, they have occurred. And the first one coincided with the increase in the frequency of severe cases of PCV2 systemic disease. And this was one of the issues that promotes a thinking line on the possibility that PCV2B could be more pathogenic than PCV2A. But the truth is that when you are comparing them under experimental settings able to reproduce the disease, those differences are not clear. So the real cause of this genotype shift from A to B is really not understood. Might be PCV2B could have better fitness and replicate better in piglets than A. Well, this is again speculative, but we do not really have the answer for that. The same happened with the second genotype shift, because when we start vaccinating in a massive way, which was starting in 2008, 9, 10 especially, then we observed this genotype shift around 2012, 13, etc. and coincide with this advent of massive vaccination. Again, one may say, might be vaccines are able to, I wouldn't say protect, but might be react better against PCV2B than D. Even D is a kind of evolution from PCV2B. So from this point of view, this is a quite potential explanation. So the immunological base here would be very important. But again, it has not been fully demonstrated because the PCV2 vaccines are based mostly in A, and they are protecting against B, and they are protecting against D. So a kind of genetic or immunological advantage should be present. However, to date, we have not been able to demonstrate, at least in a clear-cut form.
Right. Have we any evidence at all, Kim, that we've allowed certain genotypes to evade or escape the immunity caused by vaccination? Have we favoured somehow these sorts?
Well, what we know nowadays is that there are some antigenic differences between the genotypes. And if you are working with monoclonal antibodies, you are able to find monoclonal antibodies that are able to neutralize one genotype and not another. So from an immunological point of view, there are differences among genotypes. However, in general terms, when there's a natural infection, you have a polyclonal response from a humoral point of view, and you have also cellular immune response. So what we believe nowadays is that those changes in particular epitopes are not sufficient to avoid the protection or the immune protection provided by the vaccine so far. Of course, we do not really know if in the future we can create or escape mutants can be created spontaneously. So far, I believe, do not exist, and we have never ever demonstrated so far their existence to date. For the reason it's important to monitor PCV2 and PCV2 sequences, because it's very important to know what's going on at each time, which genotype is the most prevalent, and if there's a kind of genotype shift, especially thinking on a potential new genotype not able to be protected sufficiently by the vaccines. However, if this happens, the most logical way of thinking is that we should observe something at a clinical level. And I would like to back importantly to that part, because I insist, it looks like that we are focusing a lot on the virus, on the genotypes, on the mutations, on the potential genotype shifts, but who is telling us the answer is the pig. The pig should tell you how they feel, how they produce, how they grew, etc., etc. This is the most important issue that will tell us that something is going on. Remember, back into the late 90s, we were in front of a new disease because pigs told us, hey, I'm suffering wasting, I have some other lesions that I have never seen before, and it's not PRRS, it's not other diseases which are known, etc. So we should expect a kind of clinical signal as well. If we are just focusing on the virus exclusively and not with the host, probably we are wrong. We have to follow up both at the same time.
Is the pig telling us at the moment that it's being affected differently by PCV3 than by PCV2?
Well, PCV3 it's again a quite ubiquitous virus, or at least is worldwide distributed, but what it's able to cause PCV3 is not entirely clear. Let me explain. In fact, there are a number of groups in the world that they have been publishing information about PCV3, and you will see a number of scientific papers in which say PCV3 associated with a particular condition. In most of the cases, this word associated is probably understood wrongly, because one thing is PCV3 found in a sick pig. Okay, we can find PCV3 in a sick pig, and we can find PCV3 in a healthy pig, like PCV2. So the point here is that to find PCV2 in a pig doesn't preclude you have disease. You have to fit with the diagnostic criteria that we have been discussing elsewhere. With PCV3, we need, again, diagnostic criteria. When should we consider that PCV3 is able to cause real disease? And nowadays, we have seen that in most of the cases, PCV3 causes subclinical infections, or at least we cannot really associate clinical signs with such infection. The best way to at least propose, and I said propose that one virus is able to cause one disease is at least to see the disease associated with certain lesions and the lesions associated with a particular virus. And with PCV3, we have observed that at a reproductive level, there is a kind of association. We have observed reproductive problems, sometimes with mummies, sometimes with stillborn piglets, in which there are some lesions, mainly periarteritis, which are linked and clearly associated with PCV2, where the PCV3, where the lesions are. So it looks like that at least the reproductive stuff could be linked with PCV3 unequivocally. However, how frequent are those PCV3 reproductive problems? Nobody knows. In fact, in North America, where it has been described in a more extensive way, they shown a very nice work at Iowa State University in which several cases of PCV3 associated to lesions in those reproductive problems. And also it has been observed PCV3 in animals suffering from myocarditis as well as systemic periarteritis. For example, in nursery pigs. However, again, the frequency in which those animals, which are diseased, because the clinical signs sometimes could be confused with PCV2 systemic disease, then there's a kind of, let's say, refined association with the virus, with some lesions, and with a clinical condition. But which is the frequency of this post-winning problem associated with PCV3? We have been looking at cases here in Spain. We have never seen it. And very recently, we have observed one of those typical cases in which there's wasting, there's also a presence of periarteritis as well as myocarditis, and PCV3 in the lesions, in one case in Portugal. But it's the only case that we have seen. This means that maybe we have overlooked cases before. I don't know, retrospective studies will tell us. But it looks like that PCV3 is a pathogen, can cause disease, but we do not really know which is the frequency in which this disease is caused.
Now, with PCV2 and its association classically with PMWS, I always assumed the pig became infected from the mother, and that was really what should be focusing on. Would that be the same automatically with PCV3, or could that be something which was not maternal?
Well, in fact, the systemic disease caused by PCV2 is not necessarily linked to the infection of the sow. You can have post-winning infection and develop the disease. A different thing is that the sows have always been playing a role, because at the very end there's infection in this collective and they may infect younger animals, and this can be, of course, infecting the mates that you have in your nurseries. But you do not need to have infected sows to suffer from a PCV2 systemic disease. With PCV3, we have been observing that probably the most important clinical condition apparently associated is the reproductive one. So the infection of the sows looks like key here. What happened in these very sporadic cases of potential post-winning disease is something to be determined. I believe nowadays we do not have information enough about this condition, because at the very end it could happen that this is just a post-winning infection, or maybe it's an intrauterine infection, or something that had occurred during the lactating period. We do not really have any clue on that. We have to think that with PCV3 we are in the very beginning of potential association of the virus with the disease, especially in terms of diagnosing disease in a let's say more routinary form.
And although it doesn't evolve as same speed as you've indicated as PCV2, a diversity of genotypes for PCV3 is not an impossibility, is it? Eventually, I think we're likely to find those as well, aren't we?
That's correct. However, we have been sequencing PCV3 quite intensely during the last few years, and it's amazing. But I would say that almost all sequences that we obtain are between 97, 98, 99% identical. So this means that the degree of variation in PCV3 is very, very, very low. And so far we have proposed that exists only one PCV3 genotype. However, and when you look at the literature, you see that there are two sequences, only two, which are rather divergent, which came from China. And if those two would be found elsewhere and in a more frequency that the ones that they have been found so far, it would constitute PCV3B, so a different genotype. In fact, there's a very recent publication from this week. They pointed out that might be there are even more than those two sequences which are divergent from the current PCV3A genotype. But this is something that we have to look in the future, because as I told you, the mutation rate is much, much lower. So we do not expect the high variability that we see in PCV2 for PCV3. But of course, we have to give time on that. But probably we will not get that.
Yes. Now, time is our problem today, by the way. We're going to have to finish this shortly. I regret to say very much. I have to ask you about wild boar. I know you were due to have in Spain, weren't you, an international symposium on wild boar about the time when you and I are talking. Now, of course, wild boar have been in the headlines because of African swine fever, but I had seen somewhere that porcine circoviruses do occur in wild boar. Is this significant? Is this only PCV2 or one of the genotypes of PCV2? Where does wild boar fit into the circovirus story, please?
Yeah, in fact, the wild boar is fully susceptible to PCVs and probably all PCVs. So far, it has been demonstrated infection by PCV2, by PCV3. The percentage of infection of the wild boar may vary even by age, probably related to the generation of immunity. There are different genotypes of PCV2 that has been found in the wild boar, but we believe that the wild boar doesn't play a major role in the epidemiology of the PCVs. We must admit that PCVs are widespread in the domestic pig population, and probably the wild boar were infected because of the domestic pig and not the opposite way around, because one may speculate always that the wild boar may serve as a reservoir for a number of diseases, but take into account the prevalences that you may find in one and the other, probably wild boar doesn't represent a real risk for for the domestic swine in terms of PCVs.
Well, I'm very sorry that I'm afraid I have many more questions, but no more time. I'm sorry, Professor Segalés. Thank you very much for your help about the growing diversity of circoviruses in pigs. It's very good to hear that our vaccines are still working to help us. It's good to know that the monitoring is happening to make sure that we're aware in advance what might come along the road, but it's not yet a cause for concern as far as I can hear. It's certainly worth watching at the research level all the same. So may I say thank you indeed for your help in this podcast, and say thanks to our viewers and our listeners for joining us in this Meet the Expert episode, in which we've interviewed Professor Joaquim Segalés in Barcelona about porcine circoviruses, and even more so the newer ones, to me anyway, PCV3 and now PCV4. If we have another podcast in a year's time, we'll probably add to that list again. But for the moment, thank you for joining us. Thank you, Kim Segalés. It was a great pleasure, and goodbye. Just before you go, we hope you've enjoyed hearing our conversation with the most recent winners of the annual European PERS Research Awards. Bear in mind that next time, one of the winners could be you. Boehringer Ingelheim is again providing 3 awards of €25,000 to fund the winning PERS research studies in Europe, and is particularly interested in practical proposals. The deadline for submissions is the 1st day of July, and more information can be found on the website pers.com. We hope to hear from you.